And now we're going to focus on the cardio part of cardiovascular physiology. So starting out with some random cardiovascular facts. So there are about 100,000 kilometers of blood vessels in the body that, if they were laid end to end, would go around the world 2.5 times. So contained within your body, there are that many blood vessels. And most of that comes from the smallest blood vessels, which are called the capillaries. And the capillaries are so small and so numerous, they are next to almost every single cell inside of your body, feeding them oxygen, feeding them glucose and nutrients, picking up waste products, etc. Okay? So we'll talk about the vascular part when we talk about that later on. We'll talk about the different types of blood vessels. But there's a lot of them because we have to be able to distribute these substances around the body. The heart itself creates enough pressure in the left ventricle to squirt blood 9.1 meters. So, like to the back of the room. It's like particularly relevant as we get towards, horror movie season and stuff like that. You see people getting their heads cut off and then ejecting, etc. Right? So why is that going to be the left ventricle? Why aren't we saying the right ventricle instead? What's so important about the left ventricle? Where's the blood going from the left ventricle? Yeah, it's going to the aorta, it's going to the whole body, right? So what we'll find is, as we're learning about the heart this week, and as we're continuing, we're going to do a heart dissection towards the end of the week, we'll find that the left ventricle is much stronger than the right ventricle, because the left ventricle has to eject blood to go to the entire body, whereas the right ventricle just has to eject blood right next door to the lungs. So it's not going very far. It's going to be a key point when we're doing the dissection to look at the thickness of that wall to help us figure out which side is left and which side is right. You're starting out with models where everything's anatomically correct and is colored and painted and numbered and stuff like that. And so it's very easy to figure out right and left. But when you get that sheet part in front of you that's just like beige and yellow and stuff like that, and it's not labeled and it's different from your neighbors, etc., this is going to be the one thing that can help you to orient what you're looking at. Number one, ventricles are found down towards the apex, so we know that. But figuring out left and right, we're going to look at the thickness of the wall. The left ventricle has a much thicker wall to it. It's much stronger compared to the right ventricle because of the function for it, that it needs to eject blood a much further distance. So it's a good example of this whole relationship between structure and function, or anatomy and physiology. Capillaries, like we were talking about, that are so numerous, they're also super small. They're about 1/30th the diameter of a human hair. They are so small that individual red blood cells, white blood cells, etc., have to go through them single file, like we were talking about before. In fact, oftentimes the red blood cells will have to flex with that little indented area, will have to flex in order to squeeze through those capillaries to get to where they need to go. It takes about 1 minute for a blood cell to circulate around the entire body. So when we start talking about cardiovascular physiology, towards the end of this chapter, as well as when we do the cardiovascular physiology lab, we're going to be talking about the amount of pressure that it exerts, the flow rate, and things like that. How fast can we get this blood to circulate around the body? In general, it goes a full circuit in 1 minute. So it's a pretty efficient system. So the human heart rate, about 100,000 beats per day. Now, on exam day, that's significantly higher. But on average, it's about 100,000 beats per day. So it has this natural rhythm to it. It's always beating. It's the pump that drives the flow around the system. If that stops, then everything else is going to be affected, right? So it's going to be very important that it can continue on. Okay, so we're going to look at some basic anatomy for, again, the cardio and then the cardiovascular system itself. We have these different circuits or pathways that lead to different parts of the body. The systemic circuit is going to be going to and from the body itself, to the head, to the arms, to the lower limb, to the torso, etc. The pulmonary circuit is going to be going to and from the lungs. Okay? There's an additional circuit, a third circuit. Early lab people, what's the third circuit? Yeah, the coronary or the cardiac circulation. These are blood vessels that are just going to and from the heart itself, making sure that it stays alive and gets what it needs. Okay? So, when we look at the heart, we're going to see a series of blood vessels entering and exiting the heart, distributing the blood to these three different circuits: systemic circuit, pulmonary circuit, and cardiac or coronary circuit. Now, one of the things that we see in terms of the mammalian and human heart is that it's divided in various ways. So it's divided between atria and ventricles. Atria are going to be located towards the top, the superior chambers. Ventricles are down below. And it is separated between a left and a right side. So there are walls, intraatrial walls and interventricular walls that separate those chambers. So what we have is that the right side of the heart receives deoxygenated blood, okay? Quote unquote blue blood, right? So we're pooling that deoxygenated blood like from the body after we have delivered oxygen all throughout that last minute. We're pooling it along the right side, and then we're sending it, bless you, to the lungs to exchange that oxygen and carbon dioxide. Afterwards, it's returning from the lungs. It goes into the left side, goes into the left atrium, which then sends it down to the left ventricle. Left ventricle sends this red, highly oxygenated blood out to the body as well as out to the coronary circuit. Okay? So we're separating our low octane and our high octane gas, our deoxygenated blood on the right and our oxygenated blood on the left, so that what goes out to the body has as much oxygen as possible. We don't want to mix it and get some sort of deoxygenated mix going on there, right? We have a lot of cells. These cells are really active. They need that oxygen to make their ATP, to maintain our internal temperature, and so we need to make sure it's as highly oxygenated as possible, okay? So they see that, you see that in endothermic organisms, like warm-blooded organisms like mammals. They have that separation between the two sides, and they have two atria and two ventricles. If you go to different organisms, their heart structure is totally different. So for instance, you have simple invertebrates that are, again, ectothermic, cold-blooded. They have simple circulatory systems. Some of them will have open circulatory systems. Rather than keeping the blood inside of all the blood vessels like ours, the blood, there may be a pump, like a heart, or maybe multiple hearts, and that moves it, but then it kind of rains down on the relatively simple organs inside the animal, and that's how it distributes the oxygen and the nutrients and stuff like that. It doesn't have directed blood vessels working it everywhere. It just kind of like a sprinkler system rains down the organ. It collects kind of along the bottom side of the organism, routes it to the heart again, and the heart sprays it out onto the organ. So it's called an open circulatory system. So a very simple pump. It collects it and it ejects it again. You have things like fish. Fish tend to have two chamber hearts. It has an atrium, which receives it back from the body, and then it has a ventricle, which is the forceful part that pumps it over to the gills to be oxygenated again. And then from the gills, it goes out to the body. So just two system, or two chamber. You go to amphibians, they have a three-chamber heart. That means out of our four, one of them is missing. Two atria that are actually collecting it from the two different circuits, but it pools inside of one ventricle. So when that ventricle contracts, it's sending some of it to the lungs and some of it to the body, but again, it's kind of a mixed octane. But amphibians, cold-blooded, etc., their cells don't need as much oxygen as ours do when we're using aerobic respiration to help generate our body heat and keep our bodies warm. Okay? Okay. So, more than you maybe wanted to know about hearts and things like that, but still kind of interesting facts that you can impress your friends with. If you have squid and things like that, they have 3 hearts, different hearts. They're very lovable organisms. Lots of arms for hugging and 3 hearts. They should be on every single Valentine's Day card, right? It's a good example. Okay, so like we said, these are our circuits that we see here. We see 2 different sides, right? And 2 different chambers. It separates like that. Atria at the top, ventricles at the bottom towards the apex of the heart, atria up towards the base where the blood vessels are entering and exiting the heart, right side deoxygenated blood, left side oxygenated blood, pulmonary circuit to and from the lungs, systemic circuit to and from the body. We also said that there's a third smaller one, which is the coronary or cardiac circuit, right? Now, one of the things that we're seeing here that we're noticing in the lab as we're learning here is that we're seeing arteries and veins, right? Arteries always take blood away from the heart. Veins always take blood to the heart. So when we say the aorta, that's the largest artery in the body. It's taking blood away from the heart. Veins, like the vena cava, veins are taking the deoxygenated blood, in this case, back to the heart. So veins always return back to the heart. Arteries go away. When we look here at the pulmonary circuit, it gets a little bit weird. Because arteries here, this makes sense, arteries are red, oxygenated blood, veins blue. But in the pulmonary circuit, it's the opposite, okay? Here, pulmonary arteries, because the blood is going away from the heart. That's the definition of an artery. All arteries go away, okay? But notice that it's blue because that's deoxygenated blood going to the lungs. The whole reason we're going to the lungs is to pick up fresh oxygen and get rid of carbon dioxide. So in the pulmonary circuit, like we see up here, the coloration that you would normally expect for arteries and veins is backwards, okay? That also occurs in what we'll talk about, the fetal circulation as well, okay? So pulmonary circuit, we still have arteries going away from the heart and then veins coming back to the heart, but they have an opposite coloration that we would expect. All right, other things. For heart anatomy, we know that it sits within the thoracic cavity. It sits within its own pericardial cavity as well. So it is medial to our two lungs. It is superior to this big muscle here, which is called the diaphragm which ventilates the lungs, okay? We see, again, the base is up at the top here. The apex is down towards the bottom. And we can see that it actually sits inside of a bag. So there's a bag here called the fibrous pericardium, or it's called the pericardial sac. So that creates an environment that the heart sits within. There's some pericardial fluid in there as well, not a lot, but some. And lining the inside of that sac is a serous membrane that's called the parietal pericardium. So you can call it the parietal pericardium. More specifically, they call it the parietal layer of the serous pericardium, but you can just call it parietal pericardium. So it lines the bag, and then if we follow that around, we can see that there is another layer of it that's right on the surface of the heart, which is called the visceral pericardium instead, or the visceral layer of the serous pericardium. So these are slippery serous membranes that we learned about in the beginning of 201, right? We had mucous membranes, we have serous membranes. Serous membranes line the outsides of organs and line the cavity that they're sitting within, so that when they're moving around doing their jobs, they're not generating friction on each other, right? When you look at the torso models, we have a ton of different organs packed in there, intestines and all kinds of other organs right next to them. And so when they're moving, like the heart moving next to the lungs, we don't want them generating friction. So we have a visceral serous membrane that's right on the organ, and we have a parietal membrane that lines the cavity, okay? So visceral means gut, so we call this one a visceral pericardium, right? Anything you see cardia refers to heart. Visceral pericardium on the surface of the heart, parietal pericardium lining that pericardial sac, okay? In addition to these membranes, we have 1, 2, 3 layers of the heart, okay? The innermost layer here is called the endocardium, and this is a epithelial lining that lines the insides of the chambers. So in the blood, remember, as we were talking about the clotting cascade, the blood usually only sees epithelium. That's what its norm is, okay? Whether it's traveling in the blood vessels or whether it's entering the heart itself. If it sees anything else, like collagen fibers or something like that, or muscle, it starts attaching to it and clotting on it. So we got to make sure that we have a nice epithelial lining inside the chambers too. So inside the heart, that epithelial lining is called an endocardium. That's the innermost layer of the heart. Then the middle layer of the heart is called a myocardium. So myo means muscle, and this is where all that cardiac muscle tissue or those cardiac muscle cells are going to be found. It's the thickest layer of the heart. It provides the action for the pumping of the heart. Then the third layer is called an epicardium. Now this is where it gets a little bit confusing because the epicardium is the outermost layer of the heart, but structurally, you see how it's in parentheses right here? That membrane, which is right on the surface of the heart, like we said, like saran wrap, that's the epicardium. That's the outermost layer of the heart. So it has two different names. If we're talking about it as a layer of the heart, we call that the epicardium. If we're talking about it as a membrane of the heart, then we call it the visceral pericardium. It's the same exact structure. It's a serous membrane on the surface of the heart like saran wrap. It counts as a membrane. It also counts as one of 3 layers of the heart, the outermost layer of the heart. It just has 2 different names. Okay? So 3 different layers of the heart: endocardium, myocardium, and epicardium. Okay. So like we said, we don't want friction on the heart. We want the heart to move very smoothly. That's why we have these serous membranes. They secrete a pericardial fluid. If there is inflammation of that pericardium, swelling, then you start to get friction, right? So you can actually get pericarditis, right? Itis, anytime you hear itis, that means swelling, arthritis, swelling of the joints, right? Hepatitis, swelling of the liver due to various things. Could be viral, could be abuse, etc. But pericarditis is where we lost that nice smooth movement of the heart anymore. Okay, so if you start to generate friction or, for instance, excess fluids there, it can affect the movement of the heart. Now it's generating friction or there's excess fluid, so it doesn't move quite as easily. Again, when I'm going through this, I'll usually bring in examples of how the structures we are learning about relate to maybe diseases or disorders that you might see in your career. But in general, I'm not going to test you on diseases, disorders. You get a whole nursing program or dental hygiene program or something like that. That's where you're going to be focusing. We're laying the foundation now. How does it work normally? What do the parts look like normally? Okay? So we're just giving it to you so that it's familiar to you, but in most cases, unless I specifically say you should know about this, like anemia or something like that, you should know the definition of anemia. But other than that, this is just for your interest, okay? So don't worry about these ones that talk about clinicals or homeostatic imbalances instead. All right, so there are three layers, like we mentioned, epicardium, myocardium, and endocardium. The myocardium itself, we said, is the thickest layer, and that's where our cardiac muscle tissue is going to be located. So it's the thickest layer. In fact, it's thicker, like we said, on the left side compared to the right side, and it has an interesting pattern to it as well, where we have this kind of winding pattern of cardiac muscle bundles that totally surround the different chambers. Because the whole idea for this cardiac muscle is that it can squeeze the chamber, okay? So by squeezing the chamber, we're working to compress the chamber and eject the blood from it, okay? So imagine, if you will, that you are in a chamber right here, right? This might be like an atrium. So all of you guys have pooled here in the atrium, right? And imagine, if you will, that the walls all of a sudden started coming in, right? All the walls start coming in, you start feeling some pressure because there's less and less air maybe to breathe and there's less and less space to be. And so what you might want to do, as an example, is you might want to leave, right? Because all of a sudden you're like, oh, I'm really getting stressed out in here, so I'm going to leave. And so with that decrease in volume as the walls are coming in, there's an increase in pressure, right? You're bumping into each other, etc. And so you will tend to leave. And what will happen then is, for instance, as that cardiac muscle squeezes the atria, the blood goes wherever it can to eject, and it goes through those atrioventricular valves and drops down into the ventricles, okay? Likewise, when the blood is now at the ventricles and we start constricting the walls of the ventricles, that's going to put a squeeze on the blood there. The blood's going to try to exit out whichever way it can, and it will go from the ventricles out those major arteries, out the pulmonary trunk of the pulmonary arteries, or out that arch of the aorta in order to escape those walls coming in like that. So the way that the muscle tissue is wrapped around these chambers gives a good squeeze to it, right? Imagine, if you will, like squeezing a sponge. It's one thing if you just like grab one little corner of the sponge, you're like, oh, I'm not getting a lot of water out. You take it, you fully wrap the sponge with your hand and you squeeze it from all sides, you get a lot of volume coming out. Same thing in terms of how this cardiac muscle wraps and encircles these different chambers. Okay, so in terms of heart anatomy, that's one of the major things that we're covering this week in lab. You're going through and you're learning a bunch of these different structures, the different chambers, different vessels of the cardiac or coronary circulation, different vessels of the pulmonary or systemic circulation, etc. So we're not going to spend a lot of time going through this because you're going to spend hours on it learning these things as we see it in the lab as well. But obviously the anatomy is important. From the surface, we tend to see, again, portions of the blood vessels and the cardiac circulation. We see those ear-like flaps that we call auricles, which allow a little bit more volume for the underlying atria. When we look at the posterior side of the heart, we can see again where some of those blood vessels are entering and exiting. We can see that all important, what's called coronary sinus. The coronary sinus is a major draining point for all the deoxygenated blood that has served the heart and delivered the oxygen and the nutrients, and now we need to pool it back together again. So this area where you can see is a bit bigger diameter is the coronary sinus. That dumps back into the heart. Now we have 4 chambers in the heart, 2 atria, 2 ventricles, left side, right side. This is deoxygenated blood that is returning to the heart. So of those 4 chambers, where do you think it's going to go? So it begins with A, but what's that chamber called? Atria. And it's going to go into the right atrium. The right atrium is where we're collecting deoxygenated blood. Right side's deoxygenated, left side oxygenated. So this is deoxygenated blood coming from the coronary circuit. So it makes sense that it collects in that pooling point in the right atrium so it can join the rest of the deoxygenated blood going down to the right ventricle, going to the lungs to pick up a fresh load of oxygen. Okay? Okay, so you're going to see more of that as we get into it in lab. So this is internal. Internal, we start to see those different chambers like we mentioned. We also see the different valves. So 4 different valves. Okay? We have atrioventricular valves that separate the atria from the ventricles. One on the left and one on the right. These have these little heartstrings attached, which are called what? Chordae tendineae. Those are anchored to bundles of muscle that are called papillary muscles. Okay? So for the atrioventricular valves, these have the chordae tendineae attached to them, and they have these anchoring points that are called papillary muscles. The other 2 valves, the pulmonary valve and the aortic valve, they work the same way as the atrioventricular valves. They are one-way doors that allow the blood flow to go through one way but not back the other way. But they don't have those heartstrings attached to them. So the importance of the heartstrings are that the ventricles, we know, are more forceful than the atria up top. In fact, when the blood is going from the atria down to the ventricles, most of the blood, when it's moving from the atria to the ventricles, just drops straight down without the ventricles even pushing it, right? We can just use gravity, drops through the AV valve and goes into the ventricle. It's only the last like 30% or so that needs to be squeezed out to make sure we fully empty those atria and get all of it down the ventricles before the ventricles eject it, okay? So the atria don't have to be very strong. The ventricles do. So they are physically, in most cases, if you're standing up, they're working against gravity, right? They're ejecting blood this way, and particularly on the left side, it has to go a very far distance to the head, to the limbs, etc. So they are more forcefully pushing blood out of the heart, right? Maybe like 27 feet if we were ejecting it from the left ventricle, like we said. So when the blood is being squeezed in these ventricles, it is scrambling to exit that ventricle. It will, in fact, try to go back up to the atria, right? If the walls are coming in here, even if you came in this door, if that door just seems like it's closer to you or something like that, you're going to go any exit you can. So it does try to go back up through those AV valves and get to the atria. But in fact, those valves have kind of an umbrella shape to their cusps. So when the blood comes back up, it catches in those umbrella-shaped cusps, and it brings the cusps up and together, and it effectively closes the door. So it's the blood flow that actually closes the valve, and now the blood can't go that way. It has no choice but to continue on the path and go out the aortic valve or the pulmonary valve, those one-way doors which allow the exit. Okay? So that movement of the ventricle squeezing the blood and it's rushing up towards those valves, those AV valves, and slamming them closed, that's a lot of stress on them. So we need those chordae tendineae and we need those papillary muscles to be holding them down. Kind of like if you see like hot air balloons, right? Hot air balloons, we don't want them taking off without us. So we have all those ropes which attach to people or stakes that anchor them down so that they don't go where they shouldn't when we don't want them to. Same thing in terms of the AV valves. Chordae tendineae, papillary muscles will tense to make sure the valves go up but don't prolapse and go flapping back the other way. Okay? Okay, so 4 different valves. We have semilunar valves up here without chordae tendineae, and then we have atrioventricular valves that do. Now again, with the atrioventricular valves, any of the other names that you see, like bicuspid and tricuspid, those are fine to use. If you want to use those instead of atrioventricular valve, that's okay. Whatever seems right to you. Don't make up a word. Has to be an official word, but whatever official one you find is fine. Okay. Other things, we talked about atria being receiving chambers of the heart, the overlying auricles of those ear-like flaps. We'll see in lab, if you haven't already, that we have these pectinate, these muscular ridges on the inner walls of the atria. Down in the ventricles, instead, those are called trabeculae carnae, right? Muscular ridges in the ventricles, trabeculae carnae. Up in the atria are called pectinate muscles. Papillary muscles are those that are attached to the chordae tendineae that go up to the AV valves themselves. All right, So this is part of what we're going to see on Thursday when we do our dissections, that between the two ventricles, even though they're both stronger than the atria, the wall of that left ventricle is much thicker compared to the right because of the distance of which the blood in the left ventricle has to go. Okay, so sometimes in those objective sheets that we keep talking about, there may be questions where they ask you to trace the flow of blood, in this case, from one place to another. Bless you. So when we're talking about tracing the flow of blood, we're talking about you being able to do something like this, like in your mind being able to follow a blood cell from the time it exits or enters the heart to the time maybe it exits to go to the lungs. So passing through the different chambers in sequence, past the different valves, etc., right? So receiving the blood back to the right atrium through the dry cuspid valve, down the right ventricle, from the right ventricle through the pulmonary valve to the pulmonary arteries to the lungs. From the lungs, we're going to return via pulmonary veins, dump into the left atrium, past the left AV valve or bicuspid valve, left ventricle to the aortic semilunar valve, and then out the aorta to go to the system. Okay? You've got to be able to visualize that flow. Now, the interesting thing about this is we can only envision this one step at a time, but in truth, the way that the heart contracts, both atria, right and left, and both ventricles are contracting at the same time. Both atria contract at the same time, both ventricles contract at the same time. So it goes atria, ventricles, atria, ventricles, atria, ventricles. When the atria are contracting, they are sending the blood down to the ventricles. The ventricles necessarily need to be relaxed so that they can fill and receive that blood. Once the atria are done contracting, the ventricles have received all their blood. Now it's their turn to contract and eject that blood. When the ventricles are ejecting that blood out those arteries, the atria are in their relaxing and filling stage, getting reloaded for the next cardiac cycle. Okay? So it's atria, ventricles, back and forth, back and forth. So even though we're diagramming it out like right atrium does this and left atrium does that, it's right atrium and left atrium are both sending blood down to the right and left ventricles. Right and left ventricles are both ejecting blood out of those major arteries at the same time. Okay? Okay. We mentioned coronary circulation as well. You're going to identify some arteries, the main ones being the right coronary artery and the left coronary artery, and then some of the branches that come off of that. There are veins as well, like we said. Great cardiac vein is the biggest one, starts on the anterior side, curves all the way around to the back on the left side. There's also a middle cardiac vein that we see here. On the posterior side, there's a small cardiac vein that you don't have to know, but it handles the right side. All of them pool, like we mentioned, there inside the coronary sinus. So coronary circulation or cardiac circulation is keeping the heart itself alive. So we mentioned the heart valves. This is what they look like in terms of a horizontal cut. You can actually look down at them, and you can see all of them have that same type of umbrella-shaped cusps. But with these ones, they call these semilunar because they kind of thought that they look like crescent moons, if you will. They call this one over here the tricuspid valve because there is 1, 2, 3 cusps to it, whereas the one that's on the left-hand side is the bicuspid valve because it only has 2 of those umbrella-shaped cusps instead. So that's where their names come from. And in fact, doctors, when they're trying to listen to make sure that these valves are doing their job, they can move the stethoscope around to different areas around your chest, and it tends to highlight each of those 4 valves so that they can listen for that common staccato type of sound, the bump, 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 bump. That has to do with the valves closing each time as the blood rushes towards them. So if you have a valve that is not closing all the way, like the flaps aren't coming together all the way, they've become kind of stiff and not flexible, or they're flapping backwards, you're not going to get that nice sharp sound, the bump, 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 bump. You might get a bump, shh, a softer kind of whispering sound because some of that blood is actually leaking backwards instead. Okay, so this is just a view of, like we said, some of those papillary muscles attached to the chordae tendineae going up to those valves. You can see it here as well. This is the type of view that we're going to see on the sheep heart on Thursday. Again, look at the thickness of the left side, or the left ventricle, compared to the thickness of the right side. A big difference there that we're going to highlight. Okay, so this is just a graphic that goes through what I was talking about in terms of these being one-way valves. that are allowing the blood to flow through the heart in one direction. So AV valve is allowed to go from the atrium down to the ventricle. If the blood, and when the blood, decides to go back the other way, it fills these cusps that you see over on the side there. So it comes up and catches inside of these cusps and actually causes them to close. Okay? So they are what drive the closing of the valve. And then, like we said, once it can't get out this way, it has no choice but to then go out this other one-way door and exit through those different semilunar valves instead. So we can see how to blow these up. These are one-way doors, but they're oriented the correct way. So we can push on them this way and have them open. But if it tries to come back the other way, it's going to fill those cusps and close the valve. So it's much like the one-way doors we have here. As you're exiting today, you're going to push on the door, it'll open. If you try on the other side to push on the door, everybody's going to make fun of you. But if you try to push on the other side, it's a one-way door. You're just going to be sitting there going like this, right? In fact, if you're pushing on the other side of the door, it's going to help to close the door. Same thing in terms of these valves. So if the blood's pushing from one side, it will open the door. If it tries to push from the other side, it effectively closes that door. So the blood has one way to go. So like we said, in some cases, those valves will not be working. If you have an incompetent valve where you get some of that backflow, or you have stenosis where the flaps have become stiff, then what they will do is they may replace the valve. They may put like a pig valve in there. They may put a synthetic valve in there as well, and or a cadaver valve. And if they do something like that, remember we talked about the whole clotting cascade? That's foreign tissue, right? So the whole clotting cascade could start attaching to it. So generally people have had valve replacement. They're also on something like warfarin or Coumadin. They're also on medicines to make sure that their immune system, those white blood cells, don't start attacking that tissue. So it's a trade-off, but having a functioning heart is more important than having to take these pills on a daily basis, etc. Okay. So other things associated with the heart, you will hear terms like this, angina pectoris. This is actually the pain that people will go through when there is a problem with the heart. So the angina is the pain itself. It may or may not indicate that there is death or necrosis of the tissue. Myocardial infarction, an MI, is actually a heart attack, where, for instance, you've got a thrombus that has blocked one of those coronary vessels. For instance, if we're talking about like that left coronary artery, which feeds the anterior side of the heart and the left side of the heart, etc., that's the strong side of the heart. That blood flow gets blocked. They call that the widowmaker blood vessel right there, right? If that one gets blocked, the large portion of the heart, the very important side of the heart, is going to start to die off. Cardiac muscle tissue is very active tissue, requires oxygen all the time, does not rebuild or form new cells very easily at all. For the most part, it doesn't. So if you have the death of some area of your cardiac muscle tissue, then your heart is just going to continue to be weakened through time. So that pain that you have, it could be heartburn, right? Because you had spicy Thai food or something like that, or it could actually come from the death of the underlying tissue. So the angina is the pain, but the myocardial infarction is where you actually have cell death going on. Okay, now we have to zoom down to this level. We have to look at these cardiac muscle cells. You're going to look at them in lab. You're going to look at that slide again. But we have to understand what's going on with these cells. Because if you remember in 201, when we were talking about the organization of the body, you had the chemical level down here where you had ions and molecules and things like that. Then you had cells. Cells working together gives tissues. Tissues working together gives organs. Organs working together gives organ systems. And then we have the full organism, right? So when we're talking about this heart being able to contract and conduct signals and all that type of thing, we have to understand what's going down at the cellular level to understand how the heart can do these jobs in terms of contracting. So much like when you were talking about skeletal muscle, you had to learn about that whole sliding filament mechanism in order to understand how a muscle cell contracts and then the whole muscle itself contracts, right? So we're not going to go through that whole process. We're not going to go through that whole process again in terms of, right, the actin and the myosin and the tropomyosin and stuff like that. But we do need to kind of use that as a foundation to understand about cardiac muscle cells, because there are similarities and differences, right? 3 different types of muscle tissue. You learned about skeletal muscle tissue. Now is when we learn a little bit more about cardiac muscle tissue. Now, you know it from before. It's striated. It has stripes to it based on the arrangement of those thick and thin filaments. You get that banding pattern. So in that way, it's the same. But instead of having these big, long cylinders of muscle, we have short branching cells instead that kind of taper towards the end, if you will. So they have a branching pattern to them. So you see one cell might connect to another cell like this. Okay? So when they branch and they make more connections, making more connections like your LinkedIn profile, etc., means that you can have a bigger functional unit, people that you know, that you might work with, etc. Same thing in terms of cardiac muscle cells. So instead of being an individual cell like skeletal muscle, these ones are directly linked to cells next to them, and they influence each other, like you're talking, like team building or whatever in terms of the workplace, right? So they have direct connections with adjacent cells. So they have branching, and they have special cell-to-cell connections that are called intercalated discs. And inside of the intercalated discs, like you see up the right side there, you have different cell junctions called desmosomes. Desmosomes are a bit like buttons on a shirt. Right? They hold cells together at distinct points. They're proteins that kind of knit them together, or at least button them together at distinct points. But inside of those intercalated discs, besides desmosomes, you also have gap junctions. And gap junctions are small proteins that act as tunnels that directly physically connect the cells, but they create a pathway between the cells as well. So when you were talking about stimulating skeletal muscle before, right, you had a neuron coming down that would then release a neurotransmitter that would rain down around the surface of the skeletal muscle cell. That neurotransmitter would be bound to receptors, and then there would be this chemical cascade, calcium, etc., that will relay that message inside of the skeletal muscle fiber. That'll finally reach all the different thick and thin filaments, and it will get a contraction. Okay? But we had to stimulate each and every skeletal muscle fiber. With the cardiac muscle cell, we said, with the heart itself, we want the atria both to contract all at once, then the ventricles. We've got thousands and thousands of cells lining the walls of, for instance, the atria. We can't have those ones going twitch, 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 all uncoordinated, right? If that's happening, then what's happening is you're going through fibrillation, right? You're getting all these spasmodic twitches, and you're not going to get the nice squeezing that we need to eject the blood. Okay? So we have to have them all coordinated, which is what it's mentioned on the last slide when it said that they have to act as, is it going to do that for me? No. They have to act as a functional syncytium. Okay? When you look at that word and it says sync, what does that make you think of? Justin Timberlake, that's right. No, right? But even in sync, right, you had different individual members that were working together in their dance moves so that they all looked similarly. Right? So in this case, we have cardiac muscle cells that need to work together and be contracting all at the same time. Okay? So those gap junctions that we see in the intercalated discs, when an individual skeletal or cardiac muscle cell gets stimulated, much like we talked about before, we have an action potential that's going to be generated. Sodium moving this way and potassium moving that way, just like you learned about before, like in neurons and muscle cells. In this case, rather than relying on each individual cell being stimulated, those ions and their movement can directly pass from one cell to the next cell to the next cell. So it's a bit like reindeer getting electrocuted. What? Did you guys hear about that story? Crazy story, like in, where is it, Norway or something like that, right? There's this big storm, this big thunderstorm, electrical storm. Lightning came down, big herd of reindeer. It's going to be a sad Christmas, I tell you what. Don't tell your kids. Reindeer, big herd, like, what was it, 700? 300. Okay, I don't want to make the story just like 300 is still a lot, right? Lightning came down. They were so closely connected. Probably there was also some conduction going through the ground. It was wet, etc. 300 reindeer dead, boom, like that, all across the hillside just from this lightning strike. Okay? So that's a sad story about connectivity between individuals. In a happier story about connectivity in individuals, these cardiac muscle cells will have direct conduction. So one lights up and starts contracting, that immediately, almost immediately spreads to all the rest of them. So that all the cells lining those atria will contract at one time. Then all the cells lining the ventricles will contract at the next time. So they're working together in sync as one particular unit. Yeah.
And that's because of the gap junctions?
That's because of the gap junctions. The gap junctions allow the movement of these ions, of these charged particles, to move directly from one cell to another cell, rather than having to relay and have these kind of slower processes like we saw before. Okay? All right, so these actual slides that we see here, just talking about the anatomy of them, the stripe aspect of it as well. What we're going to get into as we start the next time is talking about the cardiac muscle cells and the fact that there are two different types of cardiac muscle cells. Oh no, did not see that coming. There are contractile cells which are doing the job. These are the worker cells causing the atrium and the ventricles to contract. But then there are these autorhythmic cells that are like the elite, right? The 1% that are in charge of all the other ones. So we need to talk about those two different types of cardiac muscle cells and what they're doing in the heart to make sure that it works properly. Okay? So that's your cliffhanger until next time. If you want to learn more about that, you can read it in the book or something. But we'll start there next time.